Thank you, Dr. Durham. Before I begin, I would just like to thank and acknowledge a few people and organizations. I'd like to thank the Lubbock Christian University's Department of Natural Sciences, specifically my mentors, Lucy Porter and Bart Durham, the Welch Foundation, the ARCS Foundation, the Lubbock Christian University's Council on Undergraduate Research, the Lubbock Christian University's Scholars Colloquium for allowing me the opportunity and the honor to present my research here today. Now, before I begin, I believe it's important to talk about the background of playa lakes. These are unique to the southern Great Plains, specifically the southern High Plains, which covers most of West Texas. These lakes are ephemeral, meaning they're not here year-round normally. And migratory waterfowl, such as a Canada goose, come and migrate and overwinter in these lakes in the winter months. They come in the thousands. It's not uncommon to see, the entire lake surface covered by these geese. And of course, the increase in the number of animals, especially the geese, increases the amount of fecal contamination. It's been documented anywhere between 1 and 5 pounds of manure can be produced by a single goose. And so my research is part of a 3-year ongoing study to investigate the ecological impact of migratory birds on the water quality of urban playa lakes. During the course of the study, many questions have been raised, and these questions have been addressed by student-led research. Thus far, 7 different research projects have been conducted by students. These address anything from the effects of temperature on the survival of E. coli, the influences of temperature on the patterns of the abundance of E. coli, the evaluation of methods we use to determine how many fecal coliforms there are, the relationship between Canada geese numbers and E. coli numbers, the assessment of physical and chemical factors on the abundance or the amount of those E. coli, exactly how much fecal matter actually comes into these watersheds of the playa lakes. We've even had the opportunity to genetically profile this E. coli that comes from these Canada geese feces and playa lake water. My study has been the 8th. It is the antibiotic resistance of Escherichia coli isolated from Canada geese feces in urban playa lake waters. And it's unique because I've always had an interest in antibiotic resistance, and I believe it's one of the more pressing concerns coming up, and it also has allowed me to contribute back to the overall study by conducting my own research. And so the purpose of my investigation was to determine the antibiotic resistance of Escherichia coli isolated from Canada geese feces and urban playa lake water. Just some background information on Escherichia coli. It's commonly known as E. coli. It is a gram-negative rod bacteria that is found in the intestines of warm-blooded animals. And when these animals defecate, E. coli is always found in their feces. And as a side note, E. coli, when present, the likelihood of finding a true pathogen, such as Salmonella or Shigella, is also increased. And so the E. coli used in my study came from 2 sources that were obtained during January and February of 2012. The first source was from the playa lakes that we collect water from on a monthly basis during this overall study. We used 6 urban playa lakes to do this. Once the water was collected, it was processed in the Lubbock Christian University's microbiology laboratory. The second source of the E. coli isolates was from Canada geese feces. Students in the advanced zoology courses observed the geese for defecation and upon observation collected the fresh fecal samples. These were then also processed in the Lubbock Christian University's microbiology laboratory. 50 of these E. coli samples were isolated from water, and 50 of the E. coli samples were isolated from feces. In total, 100 of these samples were isolated and put into pure culture and subjected to the antibiotic testing. Each of these test tubes here, as you can see, contain 1 pure culture of E. coli. The method I used to determine antibiotic resistance is called the Kirby-Bauer disc diffusion method. It is a standard microbiological method for determining antibiotic resistance in bacteria. Each of these plates that you see here contain 1 of those cultures. It is 1 of those pure E. coli cultures on each of those plates. Now this is a picture of 1 of those plates that was subjected to the testing. The pale yellow that you see is actually the E. coli that's grown on there. At the same time, these little white antibiotic, these little discs right here were placed and incubated overnight for 24 hours at 37 degrees Celsius. This is the result. These clear areas around these discs are called zones of inhibition. This is where the antibiotics prevented the bacteria from growing. These zones can be measured, and from that measurement, based upon a standardized chart for each antibiotic, resistance can be determined. So the resistance is determined by the zones of these clear areas. This is just a close-up showing a susceptible isolate versus a resistant isolate. The picture on the left shows a E. coli isolate that was completely susceptible to ampicillin, defined by that clear zone. The picture on the right, however, shows a resistant isolate. There is no zone whatsoever around this antibiotic. And so I chose 10 antibiotics known to be effective versus gram-negative organisms. Of the 10, 5 were completely effective in preventing the growth of E. coli. This means that there was no resistance found to these 5 antibiotics. However, these other 5 were only partially effective, meaning there was resistance found. They did not prevent the growth of E. coli every single time. And so, of all the E. coli isolates, 84% showed some combination of resistance to 5 of the 10 antibiotics. That is a high amount. Only 16% were prevented from growing every single time. And that raised a question: did the antibiotics resistance determine upon where it came from? And I found no, it did not. 84% of the fecal isolates showed some combination of resistance to those 5 antibiotics, and 83% of the water isolates showed some combination of resistance to those antibiotics. There was only a difference of 1 isolate between resistances. Upon deeper examination, I observed the complete resistance of these isolates based upon the source. 9% of the fecal isolates showed resistance, complete resistance, to 1 of the 5 antibiotics, and 2% to 2 of the 5 antibiotics. For the water isolates, 13% were resistant to 1 of the 5 antibiotics, 7% to 2 of the 5 antibiotics, and 2% to 3 of the 5 antibiotics. For example, that 2% down there could be resistant to tetracycline, streptomycin, and ampicillin. And so I then examined the amount of resistance per antibiotic of all the E. coli isolates. 7% of all isolates were completely resistant to tetracycline. Now this echoes many studies that's showing an increasing trend of resistance to these broad-spectrum antibiotics such as tetracycline. 6% were resistant to streptomycin, 4% to ampicillin, and 1% to amoxicillin with clavulanic acid. And so from this, we can draw a couple conclusions. Antibiotic-resistant E. coli was identified. We did find antibiotic resistance. Secondly, antibiotic resistance was found in both fecal and lake sources. The source was not a determinant for the antibiotic resistance. Multi-drug resistance was found, and that's something to really take note of. Again, that 2% of the water isolates were resistant to 3 different antibiotics. And resistance to commonly prescribed antibiotics such as tetracycline was found. Now, tetracycline is usually a first line of defense in the treatment of bacterial infections. And so why is this study important? Why does it matter? E. coli is always an indicator of fecal contamination. If E. coli is found in a source, that source has been contaminated by fecal matter. The study has found a high incidence of antibiotic-resistant E. coli. Again, 84% of all isolates showed some combination of resistance to 5 of the 10 antibiotics. And antibiotics resistance is a growing concern, especially in the treatment of infections. Most people are becoming more aware that increasing amounts of bacteria are becoming resistant to antibiotics. And people that live out here in the West Texas area do come in contact with these urban playa lakes. And that's important to note because they are urban lakes, they do not disappear as the normal wild type playa lakes do. They're here year round. And so people can come in contact with them, with the playa lake water and the Canada geese feces that are along the shoreline directly through wading or fishing, or even indirectly such as pets, running into the water and then coming back to you. You will come in contact with this, most likely if you do live in these regions. And so I believe the most important result of this study is that antibiotic-resistant E. coli was present in both Playa Lake water and Canada geese feces, and that the presence of this antibiotic-resistant E. coli poses health concerns to people, or possibly poses health concerns to people who use these urban playa lakes recreationally. In conclusion, the studies provided insight into an area of growing public health concern and suggests the need for continued monitoring and research of these playa lakes. Thank you. If time allows, I'll be happy to take any questions.
So, Mr. Rutherford, can you tell us a little more detail about how the collection takes place?
Yes, ma'am. We actually have a little 14-foot flat-bottom aluminum boat, and we'll go out into these lakes at 3 predetermined sites that we've used GPS to mark their locations. We'll go out there with little liter bottles, little liter Nalgene bottles, and aseptically, these have been sterilized, and collect the water by placing the bottle upside down, inverting it, and it'll fill up. From there, we'll take it back to the lab and subject it to total aerobic bacterial counts, fecal coliform counts, and such.
But what about the actual feces?
Oh, the feces. The feces were collected. The students in the zoology class basically went out to these playa lakes for a while, for a couple hours, and sat around with binoculars and watched geese for a couple hours until they defecated. They went out and collected the feces from there.
Other questions?
I'm just curious, out of the antibiotics that you would use in your test, do you know which one was maybe the most recently released and how that did? Like,
The most recently.
Is it more effective than some of the old ones that have been used?
I'll go back to that slide just to. Let's see, a couple of the newer ones are tobramycin. Ciprofloxacin is a really strong antibiotic. It's one of the newer ones. Let's see, I know, again, tobramycin is usually commonly used to treat E. coli infections, urinary tract infections in the clinical setting. And so it's good to see there's not resistance to these stronger antibiotics. That's really encouraging to see that. But the ones that have been around longer, especially tetracycline, that's been around for ages, and ampicillin, there is increasing resistance in the wild to these types.
So what's the next piece of information to add to your story? What needs to be conducted next?
I believe it's important to genetically profile these isolates that have shown resistance and compare it based upon the source. Say, if we're finding antibiotic-resistant E. coli in the geese, can we connect that to the antibiotic-resistant E. coli found in the playa lakes itself? Can we say, are these geese bringing in antibiotic-resistant E. coli?
So are there other animals? So when you collect that water sample and you're getting E. coli, are we to assume that it's just from the geese, or are there other possible contaminants, contributors to that E. coli?
Oh no, it's not safe to assume that they're strictly from that, because there's resident waterfowl that use that, dogs, of course, and domesticated animals. Playa lakes around the more rural regions, especially around feedlots, just have massive amounts of influx of cattle feces, things of that nature. So the playa lakes kind of get a whole gambit of E. coli from all sources, not just the Canada geese. Oh, no, no. I would not suggest eating anything from those playa lakes, or even getting close to, going swimming in those. So.
Given the results of now 8 studies and future, has the city of Lubbock been notified? Any responses there? It seems to me that there are ethical concerns here that need to be addressed. And we have information that, to what degree does the city council or notification of that level come into play?
Well, I think it's important to note that the purpose of these studies was not to raise concern or alarm of these playa lakes. It wasn't. It was to provide undergraduate research opportunities, and I've been a part of that. And it's already been posted, city ordinances, don't swim in the Playa Lakes. I mean, it's kind of a common sense approach in that sense, because even when compared to standard methods, you just don't swim in them because it's greater than 2,700 colony forming units per milliliter. And so, I mean, the city has posted don't swim in them, and as long as we don't, as long as we follow that, I think we'll be fine.
We may, Steve, if I could answer a little bit. It's something we may bring to the attention of the city. The overall project is not finished yet. The city's graciously given us access to these. I'd prefer not to get kicked out of them before we get our project finished. But you're right, and it may be, and actually there's some things that we're studying in the broader sense of the project, such as aeration and things like that, that may actually indicate that the city could do some things to help mitigate these problems.
Yeah,
So
because your research demonstrates that it's not just the water.
Mm-hmm.
And so swimming in the posted sign, swimming in the water is one thing, but you potentially exposed to E. coli by walking on the beach.
Oh,
So,
around that, I mean, just to kind of answer that in a way, I mean, we all have E. coli within us. We're all warm-blooded animals, and most E. coli in itself is not a true pathogen. It does not cause disease upon infection every time. And so, the off chance of a pathogenic strain is not very likely, because one, it's also very diluted in the Playa Lakes. It's a larger surface instead of coming into direct contact with that. If that kind of answers it.
Keegan?
In light of the, well, first, what sort of an incidence of E. coli infection, because it sometimes can be pathogenic, what sort of incidence rates do we see in the
Honestly, I don't have the information from that. I would have to go probably talk to the Health Sciences Center and the Covenant UMC, but it's not. I don't believe it's a very high infection rate, if that makes sense, from wild sources.
This may be a stretch, so you just correct me here, but in the city, we've received warnings about not dumping, like opening bottles of antibiotics and leftover medicine and dumping it into the water supply, flushing it or washing it down. Do you think those drugs that we put into our water supply have any effect on the increase of antibiotic resistance? Is that possible?
I mean, I believe that was the premise of most of these huge USDA research. I mean, we've observed increases in antibiotic resistance in wild sources since the 1970s. It's just been increasing. The more we use antibiotics, the more resistance there will be. And most people really don't complete their course of antibiotics. And so somehow they do get introduced to the environment. And that, I mean, logically, that would lead to an increase of resistance.
what's your publication strategy for this?
Honestly, it's part of the overall ecological impact of the migratory birds. And if I'm able to maybe continue into the genetic profiling of these resistant E. coli, publication may come from that. But most importantly, it's coming from that large overall study that this has been a part of.
Yeah, we've got a series of questions that we will answer related to this. One is we don't even know what the seasonal patterns of these are. So there's one aspect that we can look at. And then certainly the genetic profiling. And the reason we're doing the genetic profiling is to address Dr. Laughlin's question to say if we can show that the E. coli strain that's coming out of the feces from the geese that we actually go and observe, and so we know what strains are in the geese, if we can match that genetically to the strains that we're finding in the water, then you can show a cause and effect. But until we can do that, you have to assume there's other sources in there as well. And so right now there's about 3 to 4 parts of this project that we see as publishable in the larger scope. There's also one of our other students that competed for this award today did work on temperature and survival of E. coli. So the geese are here in the winter. If they defecate on the shore in the winter, and that sits there in very cold temperatures, is that E. coli still viable if it gets into the water later? And so there are pieces of this. I see probably about 5 publications that we should be pretty comfortable with getting out of this 3-year project. See a hand in the back.
Honestly, there's really not that many, because I mean they come in the thousands and they'll roost in any playa lake that they see. And actually, the playa lakes I used are part of this overall study. We used 6 urban playa lakes within Lubbock, and one of them was in Wolfforth. And so it was selected upon the lakes we have been studying already.
It's a really good question. It's my dream as part of this to actually do exactly what you're talking about. So if all of you would like to get your assignment to go chase geese off there for 24 hours a day and keep some geese out of there, that would be great.
Yes.
Any other questions? Thank you, Nolan.